The body contains over 600 muscles, which comprise about 40% of an adult's body weight. Muscles are a unique form of tissue that transform energy into motion. Everything your mind conceives is transmitted to your muscles to perform. The words you speak, the expression on your face, the motion of your fingers, as you write or play an instrument are possible only because of muscular movement. Even more, muscles operate behind the scenes to propel blood through blood vessels, drive the flow of air into and out of the lungs, digest food, and produce body heat. Indeed, this sophisticated tissue helps sustain life. This contraction of muscle can be explained through the sliding filament model. So, what is the sliding filament theory? Well, each muscle contains muscle fiber, and inside muscle fiber, there are multiple myofibrils. These myofibrils are long cylinder. Each myofibril has sections that repeat, called as sarcomere. Myofibrils consist of even finer fibers, called myofilament. There are two types of myofilaments: thick myofilament and thin myofilament. Thick myofilaments are made of a protein called myosin, while thin myofilaments consist of a protein called actin. You can remember actin as thin filament, having similarity in spelling, tin and tin. The arrangement of actin and myosin gives skeletal muscle its striated appearance. Now let's discuss the structure of actin and myosin in detail. First is the thick filament. Each thick myofilament consists of hundreds of myosin molecules stacked together, with the myosin heads facing outward. The myosin molecule, which makes up thick myofilament, is shaped like a golf club with a globular head and shaft-like tail. Other is the thin filament, consisting of two chains of the contractile protein actin. Thin myofilaments look like a string of beads. Entwined with the actin are two other proteins, called as Tropomyosin and Troponin. Troponin and Tropomyosin are two proteins that are present on the thin filaments of the muscle cells. However, they have opposite functions. This is actin, and this is myosin. Troponin promotes binding with myosin binding sites, which leads to muscle contraction. But contrary to this, Tropomyosin blocks muscle contraction by blocking binding site of myosin. You can remember function of Tropomyosin from its name as stop myosin, which means it stops the binding of actin with myosin. This is actin. This is myosin. This is N-line, while this is Z-line. Z-line is attached with actin, but N-line is attached with myosin. You can remember it as N for myosin and M for N-line. When sarcomere contracts, the thin filament will be pulled by thick filament towards the center. There is overlap of thick filament and thin filament. The Z-line move closer together. Are you feeling difficulty in remembering the structure of sarcomere? Let's understand it using an easy way. I have named it Sqadia's Guide to to the sarcomere. So, let's understand it. A sarcomere is the basic contractile unit of muscle fiber. Each sarcomere is composed of two main protein filaments, actin and myosin, which are the active structures, responsible for muscular contraction, the most popular model that describes muscular contraction is called the sliding filament theory. First, draw 2 Z-lines, which are the borders of our sarcomere. Secondly, draw a N-line in the middle. Thirdly, add a dark A band for myosin or thick filament. Finally, what's left must be the light I zone for actin or thin filament. Simple. Now, let's understand the process in detail. That basically from where it starts. In the first step, action potential opens presynaptic voltage-gated calcium channels, inducing acetylcholine release. As you can see, the release of acetylcholine from acetylcholine vesicles in second step, postsynaptic acetylcholine binding, leads to muscle cell depolarization at the motor end plate. In third step, depolarization travels over the entire muscle cell and deep into the muscle through the T-tubules. In fourth step, membrane depolarization induces conformational changes in the voltage-sensitive dihydropyridine receptor and its mechanically coupled ryanodine receptor. Then, from sarcoplasmic reticulum, calcium is released into the cytoplasm. In step 5, Tropomyosin is blocking myosin binding sites on the actin filament. Then, released calcium binds to Troponin C, shifting Tropomyosin to expose the myosin binding sites. In step 6, the myosin head binds strongly to actin, forming a cross bridge, a TP break into, ADP, and inorganic phosphate. This inorganic phosphate is then released, initiating the power stroke. In step 7, which is the power stroke, force is produced as myosin pulls on the thin filament. Muscle shortening occurs. ADP is released at the end of the power stroke. In step 8, binding of new ATP molecule causes detachment of myosin head from actin filament. Calcium is resequestered. In step 9, ATP hydrolysis into ADP and inorganic phosphate results in myosin head returning to high energy position, called as cocked. The myosin head can bind to a new site on actin to form a cross bridge if calcium remains available. In step 10, reuptake of calcium by sarcoendoplasmic reticulum calcium ATPase. That will lead to muscle relaxation. The cycle continues if ATP is available and calcium level in sarcoplasm is high. This was all about today's video. Stay tuned to sqadia.com for more precise and knowledge-stuffed videos.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.